Hello and welcome to this week's 1829 talk. You're very welcome. I am Professor Annie Tindley and it's my honour to be one of the trustees of the Natural History Society of Northumbria and to be introducing these 1829 series of talks to you. The 1829 talks highlight and celebrate the year of our foundation as a society, as one of the oldest and most honourable natural history societies in Britain. And the purpose of these talks is to allow a platform and showcase for the most exciting and cutting edge new research being undertaken by our early career researchers. So they're here really to share with us the latest findings in environmental research. As such, I hope you enjoy this week's talk. And I could urge you, if you can, to follow us on social media, Natural History Society of Northumbria, and enjoy the talk.
Hello, and thank you for joining this week's Natural History Society of Northumbria 1829 talk. I'm Johanna Weston, a PhD student here at Newcastle University, and I'm excited to share with you some of my research. I had the opportunity to present to you at the beginning of my PhD a couple years ago, so it's great to be back and share with you some of the cutting-edge research that's happening in the Hadal Zone. Tonight, I'm going to be introducing you and telling you the story of the birth of my science child, Eurythenes plasticus. While every species is special, Eurythenes plasticus is extra special because it captures the story of the plastic pollution crisis facing our oceans. I hope to leave you inspired to continue to discover new species, as well as do your part as environmental stewards. I'm a marine ecologist working on my PhD at Newcastle University. My research focuses on the scavenging amphipods who live at 6 kilometers and deeper in the Hadal Zone. I try to understand who lives there, like the supergiant in this video, and how does their low temperature, low food, and high pressure environment shape those communities. So let me introduce you to the star of the show, Eurythenes plasticus, who I've had the privilege to describe and name as part of a unique collaboration with the WWF. Please enjoy this short film that captures its discovery.
When you're diving at extreme depth, you have the chance to see something that no one else has ever seen before. There are so many discoveries to be made. If you're lucky enough to find a new species, you get to give it a name. It's a big responsibility because the name you give has the potential to last as long as the species itself. To find the right name, it helps to ask a lot of questions. What makes this species special? Where was it found? How does it move? What does it eat? The name you give must be unique, something appropriate. It should be memorable. After all, the name will go down in history.
Wow, that video moves me every time I see it when Eurythenes plasticus hits the light of the submarine. So now let me share with you and give you the behind the scenes details of how Eurythenes plasticus came to life. As you're probably aware, one of the threats facing our ocean is now well known: trash. It's estimated that 8 million tons of plastic enter the ocean every year. 80% of that are from land-based sources, entering from rivers and streams. It's clear that our actions on land matter. For the ultra-deep ocean, on the abyssal plains and in subduction trenches, we find that their remoteness doesn't shield them from plastic and trash pollution. If we look in the sediment and stomachs of animals, we can find microplastic and microfiber pieces, those that are defined as smaller than 5 millimeters. We can also see much larger and more visible pieces, like this cabling abandoned at the deepest point of the ocean, Challenger Deep. With a problem well defined, the WWF came up with an ambitious idea to draw attention to solving and stopping this problem by calling it plastic. My supervisor and collaborators at Newcastle University and I teamed up with them to make this happen. To do this, we had to first find a new deep sea species that had not been described by science. 2, find presence of microplastics in that species. 3, name it. And this is the most important step because it had to be named following the International Commission on Zoological Nomenclature in order to be scientifically valid. And 4, tell the world and share the story. Step 1 was to find a new species to science. And while you might not think that you come across a new species every day, our lab has a bit of a leg up. The ultra-deep ocean is one of the most unexplored parts of the ocean, and our lab over the last 10 years has been amassing a really large specimen collection. From previous work, we know that many of those specimens are actually undescribed species. So based on this knowledge, we zoomed in and focused on one genus, Eurythenes. This genus is composed of large scavengers that are iconic in the deep sea. They're found in every ocean, abundant, and they've been well studied over the past 40 years. But as you might have guessed from the picture, they all look very similar, or cryptic. Through DNA analysis, we're now learning that what was thought to be one species, Eurythenes gryllus, is actually multiple species. With this challenge of being cryptic, we collected multiple sets of data using an integrative taxonomic approach. Specifically, we're asking, is the identity of these specimens one of the 8 described species of Eurythenes, or does it represent a new undescribed lineage? The first approach is a morphological approach. Here, we compared how the specimen looks compared to those 8 described species. This involves selecting multiple individuals, dissecting each individual appendage, closely examining them under a microscope, looking at the details like shape and curvature of a palm, and counting the number of hairs on a mouthpiece. It's a very intimate process. Second, we extracted DNA from several specimens. PCR amplified a section called the DNA barcoding region and compared the new sequences to other sister species of Eurythenes. This tree indicates that Eurythenes plasticus sits in its own distinct lineage. With the 2 sets of data, morphology and DNA barcoding, we were confident we had found a new undescribed species of Eurythenes. In total, we had 11 specimens that were collected in 2014. The 1 male, 2 females, and 8 juveniles came from the Mariana Trench in the Pacific Ocean, and they lived 6 to 6.9 kilometers deep. With step 1 completed, it was on to looking for presence of microplastics. In 2019, our lab published a paper that showed that scavenging amphipods in hadal trenches ingest microplastics. One of those species was Hind-gut gigas, who lives at Challenger Deep. Alarmingly, every specimen of Hind-gut gigas examined had ingested microplastics, with one individual having 8 pieces. Based on that study, we were sadly confident to find it in our new Eurythenes specimens. To do this, we selected 4 of the 11 specimens, digested their hindguts, and examined for the presence of microfibers under the microscope. For this lab work, we worked in a plastic contaminant controlled lab. Any of the microfibers that we found, we analyzed them using a fancy machine called an FTIR to determine the chemical identity of the analyzed fiber. We found that the juvenile Eurythenes from 6.9 kilometers deep in the Mariana Trench had ingested a microfiber. That microfiber was 650 microns long, and FTIR analysis showed that it was 84% similar to polyethylene terephthalate, or PET. In case you're not up on your plastic polymers, PET is the primary polymer in plastic bottles and one of the top 5 plastic waste items. This work indicates that while unknown to science, it was already impacted by us. Step 3 is to name the new species. So a species is a fundamental unit of measure in biology. While you might have a new species, you just can't name it. You only get to give it a scientific name after completing a lengthy and rigorous process set by the International Commission on Zoological Nomenclature. This process aims to ensure that there is no duplication of that unit of measure. So how do you get from the data to the name? So the first step is to prepare a manuscript which presents the data, evidence for a new species, and a description of the new species. The description is based on a holotype, and here in this case it was our female from 6.8 kilometers, and it's a collection of words and pictures that allow other people to identify it. After multiple drafts, the manuscript is finally ready and it gets submitted to a scientific journal, and in this case we sent it to Zootaxa. The journal then sends it through a peer review process to 2 peer reviewers. The peer reviewers examine that manuscript, assessing for, did you make a strong case for a new species? Did you describe it correctly? And any other gaps? And while we're close to being at the name, we're not quite there yet. The reviewers send comments back to us as the authors of the manuscript. We address those comments to improve the manuscript and then send it back to Zootaxa. And this process could continue, but for us, luckily, our manuscript was then accepted, and while the name is official, there's still a few other steps that have to take place. So the first is that you deposit the holotype into a museum that's curated for people for future generations to examine the holotype. And since our specimens were from U.S. territorial waters, we deposited them at the Smithsonian. And as a biologist, it is a great honor and definite privilege to be able to deposit holotypes and type material at the Smithsonian. And the next step is to actually submit the name to the official registry of zoological nomenclature. And once that's it, we can now proclaim the name to the world. So on March 5th, 2020, our manuscript on the new species of Eurythenes from hail depths of the Mariana Trench, Pacific Ocean, was published in Zootaxa. If you wish to give it a closer look, it is available online and it is open access. Just to give you a little sneak peek of some of the figures that are in there, I'm particularly proud of the scientific illustrations that go along with creating a description. And so these are taking a bunch of microscope images and whittling down, distilling that information into drawings. And so it really harkens back to the roots, historical roots of taxonomy. And since the Natural History Society has access to so many beautiful drawings, I just thought I would share a few here. So the first one is some great photographs of the type material, and then at the bottom you can see the whole drawing of our holotype. It is also involving a lot of dissection of really small bits around the mouth, and here's some of those in scientific illustration form from the holotype. I didn't realize I was an artist until I started doing these scientific drawings, and I've never thought of myself to be an artist, but I'm quite proud of myself for how beautiful that I think they are. And then just one more, so the two limbs on the top are their ganthopods, which are what they're using to grasp food, and the next two are pleopod 4 or 3 and 4, and it's really amazing to have to be able to put down this much detail and think about a species so intimately. But again, if you want to see more, go check it out online, and it is open access. After months of hard work, we could now tell the whole world. Press releases were put out, and a 6-minute-long documentary was released that told the story of Eurythenes plasticus and the message to reduce plastic pollution. The global media picked up on our story and covered it in many different languages. Most awesomely, people have been inspired by this story and unleashed their creativity to tell the story. From posts to stop animation videos, memes, and personal redrawings, this organic global impact has been great to watch and continues to fuel my research passion. The story of Eurythenes plasticus is now being told in 2 permanent museum exhibits in Germany. In these exhibits, Eurythenes plasticus is brought up from the deep to swim in front of you. I have put in a grant to try to get a similar exhibit put up at the Great North Museum, so please do keep your fingers crossed, or get in contact with me if you have any interest in helping this along. I think it would be really amazing to have it in a museum in the UK, and especially at the Great North Museum. We've recently launched a dedicated educational website. It's filled with amazing videos and resources for learning at home or incorporating into science curriculums. Please do check it out at plasticus.school. There's a 5th step, your step, to make a positive impact. Eurythenes plasticus signals the far-reaching impact of plastic pollution on our planet. But please don't leave sad. Leave inspired to take action to help solve our created problem. And there's a few things you can do. Make sure you properly dispose of trash and recyclables so litter doesn't end up in the storm drain system and making its way to streams and rivers and ultimately the ocean. Swap single-use items for reusables, like water bottles and coffee cups. Finally, speak up at the local, national, and even international level to change how we interact with plastic goods. Finally, just want to acknowledge the full team of people that helped tell the story of Eurythenes plasticus. From science to creatives, there was a lot of different skill sets, and it couldn't have been done without each one of them. Thank you for coming to the talk. I hope that you're leaving more inspired about natural history and the deep sea, and continue to be good environmental stewards. Please do check out plasticus.school, and if you have any questions, feel free to send me an email. Thank you.